How does muscle contraction work? Let's find out with Brandon and Sarah. First of all, this is the macroscopic view of the muscle. The muscle belly is connected to the bone, and it is composed of multiple fascicles. These fascicles are made up of multiple tiny things called muscle fibers. Here, the nerve fiber, the axon, meets the muscle fiber in close proximity, separated by what is called the synaptic cleft. Here is a closer view of the synaptic cleft. When contracting a muscle, the brain sends an electrical signal through the nerve cell, creating an action potential. This causes voltage-regulated calcium channels to open, which then causes calcium to enter the nerve. Inside the nerve terminal, vesicles containing acetylcholine are present. The calcium causes these vesicles to undergo active transport and fuse with the membrane of the nerve, releasing acetylcholine into the synaptic cleft. The process of the vesicle fusing with the membrane of the nerve is called exocytosis. The acetylcholine then binds to acetylcholine receptors on the sarcolemma, causing another action potential that spreads across the sarcolemma. This causes the sarcolemma to depolarize. The action potential continues to travel across the sarcolemma and into the T-tubule. This is a closer view of the sarcoplasmic reticulum, located within the sarcolemma. And this is where the action potential continues to travel down the T-tubule. This action potential causes the sarcoplasmic reticulum within the muscle fibers to release calcium ions. The released calcium ions then bind to troponin, causing the tropomyosin to move away from the actin binding sites. This allows the myosin heads on the myosin filament to bind to the actin filament. The connection between these two is the cross bridge. To recap, the green is the troponin and the red is the tropomyosin. To bind to the actin filament, ATP binds to the myosin head and then dissociates into ADP and phosphate. This causes the myosin head to cock back into position and bind to actin. ADP and phosphate is then released and causes the myosin head to move the actin filament in a movement called the power stroke. This process continues to repeat until signaling from the motor neuron ends. As you can see here, the filaments keep pushing on each other to create more power strokes. When we zoom out of these individual actin and myosin filaments, we can see the grand picture, which is the sarcomere. The Z discs are the purple structures on the side. When the muscle contracts, the actin filaments move closer to the center of the sarcomere. When it relaxes, the actin moves away from the center and returns to its original position. The repetition of the movement of these filaments is called the sliding filament theory. When we zoom out of the sarcomere, we can see where we all began, the muscle fiber in the fascicle in the muscle belly. And that was it for How Muscle Contraction Works by Brandon and Sarah. We hope you enjoyed.